Welcome to the Global Cannabis Research Summit. We're starting with a really appropriate speaker and topic, opening doors to cannabis research, and an update from Dr. Sue Sisley. Welcome, Sue. You are a real pioneer in regard to, pushing back on the systemic, brakes that have been putting on the kind of work that you and other scientists have been trying to do here in the U.S. Under, DEA and other federal laws. So welcome.
Let me jump right in. We got a lot of material to cover, and I'm excited to be here with you guys. So Scottsdale Research Institute, our mission is to conduct randomized controlled trials to look at both safety and efficacy of smoked and vaporized cannabis. So that's it. You'll notice all of our studies look only at inhaled dry flower material, and we're studying it in three areas. We're looking at inhaled cannabis for treating pain, especially breakthrough pain associated with cancer patients. We're, of course, well known for our work in smoked cannabis for treating PTSD. And recently, we've also started collecting data on cannabis for treating opioid addiction, trying to help patients find either a substitute or a way to reduce their opioid dependence using cannabis. So this has been a lot of fun, and the reason we focus on flower over extracts and concentrates is because, first of all, flower is the least expensive version of the medicine. It has the most it still has the majority of the sales in almost all the legal markets. And that we hope that once we can get flower through the entire FDA drug development process, that it will set precedents for other botanical medicines, whether that's ibogaine or ayahuasca or whatever, that it will help teach the FDA how botanical medicines can be overseen by them and regulated. So, and of course, the ultimate goal of putting botanicals through the FDA process is that then we are able to force insurance companies to pay for botanicals the same way they do conventional pharmaceuticals. So that's why our mission is so important. And you can see this is the first study we ever did, the first clinical trial we started many years ago, trying to do this FDA phase 2 trial, looking at 4 different varieties of smoked cannabis flower in military veterans with PTSD. And you can see this was the first and only controlled trial in the world looking at this issue. There was a lot of data on cannabis for PTSD, but it was mostly case studies and observational data. It wasn't the gold standard randomized controlled trial. So we finally did finish this study in 2019. That's the binder for the 80th veteran who completed the trial. But I wanted to mention to you that the study, because of COVID-related delays and things, it's taken us a while to get the data published. But we're finally you'll probably see the data in the journal PLOS One. We specifically targeted an open source journal so people wouldn't have to pay to read this material. So hopefully you'll see but I just will disclose the results, even though I can't share all the details. I just want to give you an overall sense of the outcomes. Basically, cannabis, the cannabis that we were using in this study, the NIDA cannabis, did not show more efficacy than placebo. So it's considered a negative study. But I think we all agree that this study drug may have undermined our results. And so I want to talk with you about that more, how this happens. So first, let me point out the study that I just showed you took 10 years to complete. And sadly, that is the typical timeline for randomized controlled trials looking at cannabis as a medicine in the US. If I was just doing safety studies on cannabis, looking at harmful effects or addiction potential, those studies are expedited much more quickly. But if you dare say you want to look at the benefits, the therapeutic benefits, all of these boxes, we put together this schematic so you could see all of these boxes represent different different regulatory hurdles that you have to go through. And that's why you can see, you know, all the layers of government red tape involved in doing this work. And this is one of the most onerous obstacles, I would say, is trying to get to score this DEA Schedule 1 license. Many scientists call this the golden ticket. And we've had ours renewed 6 times now. Here's an example of our most recent. This you can see, as I cross out the number and the street address because the DEA is concerned that somebody may actually try to steal this study drug from us, and they don't want anyone to know where this is being held. This is our example of our analytical lab license. So the DEA offers several different categories of Schedule 1 license. You can have an analytical lab, you can have the researcher license. The one license that you can't get right now is the manufacturer license, and that's what I want to talk with you about. The purpose of our litigation is, you can see after we got this, we immediately moved to order, 10 kilos of cannabis, which was the requirement, what our study needed to be executed. And you think it would like arrive by armored truck or something, but there's the FedEx guy shows up at our lab door, and he has no idea he's even transporting cannabis. He's not clear on why everybody is so mystified by all this, but you can see this was our very first shipment to the lab. It was, in a cooler with a bunch of ice packs. It's shipped on ice. It's been frozen in the lockers of the research triangle for years, and it comes in these little Ziploc bags that the only information you get about this study drug is the CBD to THC ratio. That's it. They don't disclose anything else on there. And you can see this is basically, this is one example of one of the batches full of not just flower, but unfortunately a lot of extraneous plant material, stems, sticks, leaves. And you can see so it used to come in these rolled up cigarettes, and some scientists have dumped out the contents there so you can see on the top what was contained in this. So what's happened over the years, we've criticized the quality of the drug. Our concerns that the study drug is not just flour, but it's being diluted by this other extraneous material. So over the years, we've seen now they send it to us in these loose bulk bags, and it's more of a powder now. So I feel like maybe this is the government's effort to diminish our critiques is by pulverizing it into a powder. It's harder to discern the extraneous material that's in there, but it is in there. We can see it, and it's frustrating. And for those of you who aren't aware, this monopoly at the University of Mississippi was established 51 years ago. Back in 1968, the University of Mississippi was granted the only federal license to cultivate cannabis for clinical trials like ours. And the problem is, if the government doesn't like your study, they can put you into a permanent review process where you'll never, get cannabis because they're the only supplier. And that was what happened for many decades, is that it was really hard for scientists who dared to say they wanted to study cannabis as a medicine, that they would get put into this like black hole. But and one of the things I want to point out is, in order to receive the study drug, all investigators, including, this is one of our most recent versions, we have to sign a release of liability, basically saying that we take on, as the scientists, we accept full liability for any adverse events that might occur during the study, and that the government is held harmless, and the scientists have to reap all of the liability. So I thought this was insane. My attorney said, you can't sign this. This is a terrible situation. You can't sign it. But unfortunately, if you don't sign it, you don't get your study drug. You can't complete your trial. So I had to do that. I had to sign it. This was the only piece of paper that's included in the shipment. Like I said, you don't get a drug master file that tells you what are the contents of the study drug. All you get is this document, which basically tells you the THC to CBD ratio. It doesn't tell you about any other cannabinoids or terpenes or mold count or anything, but it does have this disclaimer at the bottom that says, oh, by the way, you know, after you've already signed your life away on that document, oh, by the way, this product is contaminated with mold and other microbials, and you may want to consider irradiating it before using. So I just was amazed that this has been allowed to continue this way for so many decades. When you can see here, the government, you know, that if you go on FDA.gov, and you look up what do they consider GMP requirements for the study drug, you can see under number 23 that they specifically say that there should be no mold growth on the product, and yet we know for a fact that that the study drug from NIDA commonly has excessively high levels of mold in the batches, as evidenced by, you know, when we were the first scientists to do independent testing. So we did secondary testing in an independent analytical lab, not in our current lab, where we did actually 3 separate rounds of testing confirming, you know, that most of the batches had over 100,000 CFU of mold, which would never have passed state testing in any legal market. But the government has been, you know, sending this out to us, you know, to scientists for years, and suggesting that we are required to remediate this. So I want to point out that there's 2 things that I believe are harming the outcomes of cannabis clinical trials in the US. And so we talked about the study drug. The next thing I want to talk about is the issue of fixed dosing in trials. You know, everybody's asking, how do you dose, especially in my, you know, I'm an internal medicine doctor, so my fellow physicians are always saying, well, how can we embrace this as a medicine because we don't know how to dose it? And the FDA has been forcing scientists for years to implement fixed dosing in all these clinical trials. So most of the trials you'll see, they require patients to take, you know, 20 milligrams twice a day or 50 milligrams 3 times a day, whether they need it or not. And we think that that is a huge disadvantage when it comes to, you know, being able to replicate the efficacy that patients are getting in the real world. When you force people to take a fixed dose, we found in our studies that you just create a ton of adverse events that could have been easily circumvented by allowing patients to just take what they need. You know, some patients only need a couple inhalations in these trials and they already reach a therapeutic threshold, and others need more. But the point is, everyone is unique, and if you pigeonhole people in a specific dosing, you don't achieve any good outcomes. And so we want to bring this up to say that this has to be a concerted effort from all of us in the science community to urge the FDA to rethink their paradigms when it comes to, especially to botanical medicines. It's fine if you're just doing the conventional, you know, 1 molecule targeting 1 receptor, but in this case, this is a very complex plant targeting a myriad of receptors throughout different organ systems in the human body, and we don't believe that fixed dosing is appropriate, and we're concerned that we will continue to have negative studies that don't beat placebo until we all embrace this patient self-titration model. Well, I want to emphasize that that is only when we're talking about inhaled cannabis. So I would never suggest that oral dosing should be left to self-titration. It's way too complicated. So I'm talking strictly about smoked and vaporized flower that we should allow patients to self-titrate. And now my final takeaway is about the quality of the study drug. Many of you are aware that these trichomes hold, you know, all of the goodness of this plant. You know, these are where the secretions of the cannabinoids and terpenes are the highest density in these trichomes, and yet this is what we get from our government, which is this, you know, highly over-processed plant material. And I question, what is the trichome count here? You know, when you pulverize this material, are you are there any trichomes even left, or are they all broken off and potentially dissipated in the filtering process? So my point is, what we want to study is this beautiful, frosty, intact bud material. We're trying to persuade the FDA to not force us to grind up this bud material, but instead use it as is, just like they sell in Israel or Canada, where you buy intact bud material, versus on the other side, what we are currently getting from our government, which is a diverse combination of flour diluted by all kinds of other extraneous material with less medicinal value. So let me just remind you guys how much we're all paying for this monopoly. The contract that NIDA puts out every 5 years, here's the announcement, is $68 million going to perpetuate this monopoly year after year. So I would encourage us all to try to pinpoint our energy on this effort, to try to dismantle this monopoly and finally force the government to license other growers for research, because otherwise, how do you do real world cannabis research, without being able to have access to the real world material that people are using day to day? So I think that may be, I don't know, Keith, do I have a little bit more time?
Please do, yeah.
Oh,
and
okay.
I have a couple of questions coming in, but you're on a roll, so keep rolling.
Oh, okay, yeah. Let me talk to you guys about the next study that we're kicking off here in September is a look at inhaled cannabis again, but we're looking at late stage cancer patients, and we're going to be using inhaled cannabis to treat their quality of life, their breakthrough pain, things like that. And you can see here that I'll just give you a glimpse of what this study looks like. So here are the objectives. You can see that we're going to be evaluating pain. We're going to be evaluating all kinds of other quality of life elements. So there's the primary and secondary objectives. Here is the actual material. So, we're determined to secure other sources of cannabis flower outside of University of Mississippi. And because to date our litigation has not been successful, if you want to buy cannabis in the US, your only supplier would be University of Mississippi. So the only other option, if you are determined to use something else, is you have to import it from a foreign country. And in this case, we've been targeting groups in Canada just because in order to import the flower, I have to prove that the study drug meets the FDA's GMP criteria, good manufacturing practice criteria. And Canada and maybe Israel, and there may be a couple groups in Australia, are coming close to reaching that threshold of GMP. And so that's why we've been requesting drug master files from these groups. And it looks like we found a supplier in Canada. One of the bigger LPs is going to be providing this flower, which you can see is about 9% THC, about 2% CBD. And then we have the placebo control, which is basically inactive material with just a small, very small percentage of CBD. You'll note that the placebo that we get from the government actually has no cannabinoids or terpenes in it at all. It's completely inert plant material. But in this case, the placebo, they decided to leave a little bit of CBD in. And basically this flower is going to be put into, let me see if I have that picture. Well, let me first show you the schematic of the timeline of the study design. So you can see here, this is a classic randomized controlled trial. It's going to go 4 weeks. So we're hoping that patients will have enough time to experiment with the material and gradually self-titrate to that sweet spot where they get plenty of relief without too many side effects. And I didn't want you to read, I just want to give you a glimpse of how complex why these studies are so expensive. A randomized controlled trial will cost, anywhere from $2 to $5 million for a phase 2 trial, or more than that, depending on the number of study sites. But you can see here how complicated all these tests are. You've got tons of, at each follow-up visit, there's tons of assessments that need to be completed. And here you can see that's the actual vaporizer that we'll be using for the trial. It's going to allow, the study drug to get to about 220 degrees typically. And we're going to do, sort of a modified Fulton puff method. And there you can see the recommended self-titration just for patients who may be completely naive to cannabis, that we're going to give them some guidelines on how to manage their inhalation so that they go, they ramp up gradually. And this, I just wanted to point out, is the DEA, the Schedule 1 license and the forms that need to be filled out in order to bring the cannabis material to the lab. But I raise this just because I want to emphasize that any scientists on the call who are unsure of how to obtain their Schedule 1 license to do clinical trials, we've developed a roadmap that we freely share with people. My study sponsor, MAPS, is really committed to helping scientists to be able to enter this field and not feel like it's so daunting. And so we put together tons of pages of guidelines for you for how to navigate the regulatory hurdles of the DEA so you too can get your Schedule 1 license. So let me know if you want to take a look at that, and we're happy to share that. And then this is the other study that we'll be doing afterwards. This is focused strictly on breakthrough pain in advanced cancer patients. So we're going to be comparing inhaled cannabis flower to fentanyl BUCCAL tablets. And many people think, wow, that's crazy. Fentanyl is like the nuclear bomb of the opioids. How could cannabis possibly compete with fentanyl? But surprisingly, it does, in a small pilot study that was done in Montreal, we saw that that actually inhaled cannabis performs very well because it has such an immediate onset of action versus fentanyl, which could take, from 40 minutes to an hour to kick in. Inhaled cannabis could grab hold of that pain much more quickly. It wasn't always a cure for pain, but it definitely could take the edge off the pain where people felt more functional. And so we want to build a controlled trial and collect rigorous data on how that works now that we've completed that pilot study. So I want to point out one of the interesting things that, TETRA, who is the sponsor for this next study, is, attempted to bring flower into the US a year ago. And this, you can see the press release, which I don't expect you to read, but let me just summarize for you what happened. In our effort to bring the flower in to do this study with the cancer patients, we had to submit the drug master file to the FDA, and it confirmed the presence of 3 mycotoxin in the cannabis flower. And so that's part of why we're so determined to try to end this monopoly, because if you choose not to use cannabis from NIDA, from University of Mississippi, then your option is to try to import flower from Canada. And you can see when that happens, you're dealing with contaminated material. I don't know how Health Canada didn't pick up on the maybe at that time they weren't testing for mycotoxins back then, but still, I mean, this seems like, it seems negligent that we should be forced to purchase cannabis from a foreign country when we could easily grow our own material and provide scientists other options besides NIDA. Interestingly, when this press release came out about the contamination of this cannabis from Africa, it was never, you would think that their stock would drop, would plummet. It didn't seem to have any impact, which is really interesting to me, that people seem more worried about the financial side than they do about the quality of the cannabis. But the good news is that the government did announce back in 2016 that they would end the monopoly. But here we are over 4 years later, and we still have no end in sight to this government enforced monopoly at University of Mississippi. So Keith, I'll kick it back to you.
Well, I'm so appreciative of all your efforts and the team you work with. Obviously, it's not a one human show, and yet I know there was some Q&A. So just to wrap up with some real quick ones. This was a question came online. The standardization of that self-titration is really in the pellet, and how you're allowing at some point at least to be able to know how those pellets translate into some, again, standard form. Do you think that is going to be accepted, or is that just the way, in this case, you see it optimal to deal with the fact that you're dealing with flower and self-titration? So the pelletizing is your standardization, right?
Right. This is our effort to show FDA, to give them some comfort level with flower, but I know it's also not ideal. I mean, in my perfect world, I would love to see FDA embrace bud material and recognize that, sure, there are going to be natural variations with dry bud, but that patients will self-titrate the bud, so it doesn't matter whether one bud is 12% THC and the next is 14%, because they're just going to use as much as they need to manage their symptoms. And I just wanted to show you on that screen, those are some examples of the recent litigation. Over the last 2 years, we've been suing the federal government, and you can see that we've actually persuaded the DC Circuit Court to order the DEA to respond to these 30, or actually now it's probably up to 40 applications for growing cannabis for research. And you can see here, at this point, we're on to our third lawsuit. We had a successful lawsuit last year, a FOIA lawsuit that helped us uncover this secret memo that proved that it wasn't the DEA that was blocking this. It was actually the DOJ and the Attorney General that were ordering the DEA to stand down and do not process any of these apps. So that was an important step. But again, it hasn't helped us win the war. We're still dealing with this monopoly at University of Mississippi, and nothing has changed. And that's why the litigation has become so essential.